Okay, this is my homemade neuron here at the top with these brown broken stir sticks. We have what is representative of the dendrites of a neuron. The dendrites receive chemical messages from other neurons. Moving on, we have the soma, or the cell body, which is represented by this black lid here. And the soma collects neural impulses, contains the cell nucleus, and provides life-sustaining functions for the cell. Moving down, we have the axon, which is represented here by this clear stick, and the axon transports electrical impulses to other neurons via the terminal branches. Represented by the brown cardboard wraps, here is the myelin sheath. And the myelin sheath is a layer of fatty tissue that covers the axon and ensures the signal gets there fast and with no resistance. It's like insulation for your neuron. And then here at the end, we have the terminal branches represented by the Q-tips. The terminal branches convert these impulses to chemical messages. And here at the end, I know there's not another neuron to connect it to, but the gap that would be located in between the axon terminals of the sending neuron with the dendrites or cell body of the receiving neuron is called the synapse. And neurons send neurotransmitters, which are chemical messengers that are released by the terminal branches across the synaptic gap toward the target neuron. And that is what neurotransmitters are, and this is my neuron. Okay, so now that we have gone over my lovely homemade neuron, now we are going to talk about how neurons transmit and relay the messages to other neurons. First, we're going to talk about the resting potential of a neuron, which the resting potential is around -50 to -100 millivolts, and it is when there is more sodium ions outside of the membrane and is more negatively charged. So there's more positively charged ions outside of the membrane and more potassium or negatively charged ions on the inside. It's exactly what it sounds like. It's when the neuron is resting and when it is non-excited and not firing. Next, we have the action potential, which is the firing of the neuron actually taking place. It's when neurons are stimulated with sufficient energy and called into action, which fires the neuron. This is all possible and capable from the ion channels. And the ion channels in a neuron are narrow water-filled tunnels that allow only ions of a certain size and or charge to pass through. This allows the positively charged sodium ions into the neutron, which causes the action potential. Next, we have depolarization, which is a process that happens after the positive ions flood in, moving the negative resting potential towards zero. So the neuron is building up all of those positive ions on the inside because the ion channels have opened, allowing the positive ions to flood in. So the negative resting potential is now depolarizing and moving in a positive direction. And next we have the voltage threshold, which is around -50 millivolts. And that is when the voltage controlled ion channels open rapidly, allowing positively charged ions to flood in. The neutron's voltage then surges rapidly and becomes positive as it passes zero, which leads to the action potential. So the voltage threshold is, what the positive ions have to surpass to create the action. And then after that, we have repolarization, which is when the ion flood is complete and everything kind of goes backwards. And it's when the membrane rapidly returns to its resting potential. After all of that is done and your neuron has properly fired and gone back down to its resting potential, your neurons will have a refractory period, which is a time when your neurons are either it's going to be very difficult or impossible for your neurons to fire again. But neurons only need just a few milliseconds to recharge and be ready again. So that is a summary of the structure of neurons and how they work and how they communicate with each other.